Welcome to the University of Portland campus. I'm John Watsky, Dean of the School of Education. I'm happy to welcome you to tonight's forum. It's a great turnout. We'll explore the intersection of neuroscience and education. I'm a new resident of the Portland area, and I have to tell you that I joined the university in July, and I've been very impressed with the spirit of community and partnership in service to families and education. It's been a great welcome. We look to do great things across Portland with all our schools and all our university partners. Tonight's forum is no exception. It's jointly sponsored by the Oregon Health and Science University, the Graduate School of Education, Graduate School and School of Education here at the university. Those of you in attendance represent school leaders, educators, school board members, school foundations, the research community, higher education, and the community at large. Truly an impressive spirit of community. As Dean of the School of Education, I would also describe tonight's gathering as a humbling moment as well. It's an opportunity to feature the work of others, to learn from one another, and to build alliances across our community. I know I'm doing my job as a dean when I'm humbled by the work of others, humbled by others' commitment to community. Finally, as a representative of this university, I can draw a direct line to tonight's gathering to our founding over 170 years ago in the tradition of Holy Cross. This tradition rose from the ravages of civil war in France. A small community of men and women sought to address the destruction of families and lives from social upheaval through schooling. They knew that solely academic content was not enough. This community called themselves the Congregation of Holy Cross, and their educational approach, whole child education, providing for the heart, the hands, and the mind of children and young adults. Nearly 2 centuries later, we come together as a broad community with a common interest in the holistic learner. We seek to learn more about the social, emotional, developmental, neurological, and pedagogical influences on learning in order to inform our work in support of families and students. So truly a great evening. It's with pleasure I introduce David Heil, president of David Heil and Associates, a native Oregonian, who will moderate tonight's event. David is well known throughout the country as an innovative science educator, new enterprise developer, lecturer, author, and host of the Emmy Award-winning PBS science series, Newton's Apple. Once again, a humbling moment. David was the lead author on the award-winning Discover the Wonder series, which for 5 years was the #1 selling science elementary curriculum in America. He is also the editor of the popular book Family Science. We love that book in our family. And he's working on a new publication that we all wait for, Family Engineering. In 2009, David received the award for Distinguished Service to Science Education from the National Science Teachers Association. Let's please welcome tonight's moderator, David Heil.
Thank you very much, John, and welcome to all of you. Good afternoon. Thank you for joining us for this exciting program. As John mentioned, kind of an experiment, if you will, but the right one, bringing neuroscience, education, in this case, addiction, all together and bridging those gaps and talking about them as a single unified topic with great connections. As the program suggests, you hopefully have received your program before you came in. There's a variety of pieces and steps to the program today. We're going to start with a keynote address from one of America's foremost authorities on drug addiction, Dr. Robert Hitzman. I'll introduce him to you in a moment. We're going to be followed then in the keynote address by 3 noted panelists who are sitting next to Bob Hitzman over here on the side. Let me just briefly mention who those individuals are. Sitting closest to Bob in the middle of the pack there on your left is Dr. Kathy Grant, a professor in the Department of Behavioral Neuroscience and a senior scientist in the Division of Neuroscience at Oregon Health and Science University and the Division of Neuroscience at the Oregon National Primate Research Center here in Portland, or out in Beaverton area. Next to her, on her left, is Dr. Bonnie Nagel, assistant professor in the Department of Psychiatry and Behavioral Neuroscience, who has written extensively and presented on the topics of the neuropsychological evaluation of children and adolescents. And lastly, Dr. Ellen Arwood, on my far left, your far right, is a professor here at the School of Education at the University of Portland with an international reputation in learning and language, especially as it relates to students with neurogenic disabilities. So we've got a great panel who will be responding to Bob's comments and sort of extending the topic of discussion. If you have a question that you would like to ask the panelists, in your program, there were two forms, actually. The small purple one is the one you can use to write a question down. I would ask you, since I'm the one that gets to read it, to print your question, if you would, please, so that I can read it, and pass them to the ushers who will be available and moving up and down the aisles as Bob's finishing his comment. And they can also collect those questions from you during the panelists' comments as well. And then when those questions come forward to me, I'll do my best to sort of sort through those and look for representative questions that we can put forward from all of you as an audience together. And we'll take as many as time allows us to take. Since I was pointing out the little forms, the orange one is an evaluation form. Would ask that you also take some time to complete that before you leave the auditorium this afternoon, so that we can continue to offer programs like this to our citizens here in Portland. Following the panel responses and discussion tonight, we'll continue the discussions with a general reception in the terrace room, hosted by Dr. Thomas Green, who was sitting over there a moment ago, but he's now moved. He's right here. Had to scan the audience for a bit. Who's the Assistant Provost and Dean of the Graduate School here at the University of Portland. If you did not have time to do this before the program, I would encourage you perhaps as you get up and depart for the reception to take a little bit of time to stop in at the display tables out in the foyer here at the auditorium. There are four programs represented in particular there, two large tables. There's first the Neuroeducation Postmasters program here at the University of Portland, which is one of the co-hosts for this event. And a set of two tables over here with a number of programs from Oregon Health and Sciences University. First, the Brain Lecture Series. We have one remaining lecture in the 2011 program, May 23rd, with Dr. Nora Volkow, Director of the National Institute on Drug Abuse. That you'll also find there at that table information about the BrainNet Lecture Series, which is a lunchtime lecture series here in Portland, and Brain in a Box, which is a unique program developed by the BrainNet group to reach out to schools and communities throughout the Northwest. If you're interested in more information on any of those programs, you can go to www.oregonbrains.org. The topic of the afternoon is Learning Under the Influence, Brain Implications for Children and Youth in Grades K through 12. Our keynote speaker, as I mentioned earlier, is Dr. Robert Hitzman. He's a professor and chair of the Department of Behavioral Neuroscience at OHSU. Dr. Hitzman received his undergraduate degree from Albion and did graduate studies at Wayne State University and the University of California, San Francisco. After graduate training, he taught at a variety of institutions, University of Cincinnati, State University of New York at Stony Brook, University of California, San Francisco again, and then joined OHSU, which occurred in part, I've heard him tell this story, so I'm going to share it, hopefully not trumping anything he says later. He came to OHSU in part because we had the unfortunate distinction here in Oregon of having, if not the highest, at least a relatively high number of methamphetamine addicts. It's an interesting recruiting pitch, I got to admit, but it worked because Bob came, because he had a ready body of patients that he could work with from a research standpoint. It was fortuitous for us in Oregon here, and OHSU in particular, because since his arrival, he's established not one, but two internationally recognized centers in his areas of research, particularly the neuroscience of alcohol and methamphetamine abuse. Specifically, his research focuses on how genes regulate complex behaviors, particularly complex drug-induced behaviors. I also want to mention that in addition to his research, Dr. Hitzman has also been a very strong supporter of public outreach activities just like this. He's participated in numerous brain awareness activities over the years, and earlier this month he presented the prestigious Markham Hill Lecture for OHSU, where he chronicled his 40 years in addiction research. Please help me in welcoming Dr. Bob Hitzman.
Welcome. Thank you for that nice applause. Well, I guess I have to start it myself. Your job. It's a bad way to begin any talk with a disclaimer, but I'm going to do that. The first, no.
Which one are you?
With
They're
a
not
top.
in order, I apologize. What's the name of your presentation?
One of these. That one.
Sorry, I thought they were in order.
That's okay. A disclaimer. The first is that the topic that we're going to discuss tonight is how drugs affect the brain of young adults and preteens. This is not my area of expertise. It really falls more on the other members of the panel to discuss that, and especially Dr. Nagel. What I do want to talk tonight about, though, is some of the effects that drugs do have on the brain. And for those of you that heard me talk before, you're going to see some of the same slides. I only have a limited number, and so you're going to see them. But I think it is important. We have no real reason to believe that what happens to the brain in largely people I've studied are young adults past puberty. We have no reason to believe that those changes that occur in the brain don't also occur in younger individuals. And in point of fact, they may actually be more severe in that group. So that's something I hope that we can discuss. I think no, that's wrong. Pull it down. Back. That way. Okay, so to this group, I don't think this slide needs to be explained very much. This simply is an MRI study looking at the functional activity in the brain from about age 5 to age 20. And the point to be made here, of course, is something that you all know, is that the brain changes dramatically over this period, and it changes especially around the peripubertal period, where we see a great deal of synaptic pruning and changes that are going on that lead us to have the adult brain. And so the real question we need to address in this very dynamic process that's going on, and the one that actually is very important for our maturation, how has this changed, and how has that trajectory changed if we're using drugs? And it's likely to change in different ways depending upon the drugs that are abused. And I hope that'll be one of the topics that we'll discuss. This is just a slide. I like to have a slide like this, and I've used different variations of this. What we're really interested in here is what's in the middle, is how drugs affect development. The point to be made, though, is that there are lots of things that enter into that equation. I know a fair amount about this over here, physiology, a little bit less about behavior, but we have panel members that know more about that than I do. I know a fair amount about genetics. And of course, the really important one is down here, the environment and peer influences. All those things combine to form what we might call the addictive personality, and we need to understand that in its total context. To just pick out the genetics or the physiology is really a mistake. And so I hope that that theme comes through as I continue the talk. Oh, wrong direction. I always hate doing this, tables which almost no one can read. But the top one I think is important. I'm just going to pull, here's self-inflicted injury. What's being actually recorded in this table are visits to emergency rooms. And this is the diagnosis that they had at the emergency room, and this is the sample that they looked at. And the point is, let's just look self-inflicted injuries. There's a fair number of group here. It turns out that 27% of those in the 12 to 15 group were legally drunk when they made it to the emergency room. If you go ahead all the way up to those at the age of 19 to 24, beyond the group that I think we're really interested in, half of them were legally drunk. And so the point is that many of the things that we see, visits to the emergency room, are associated with consumption of large amounts of alcohol. And not shown in this table, and they weren't tested for it, it's likely that other drugs are on board as well. Another statistic which I think is frightening, this is from 2003, there are about 1.5 million 12 to 17-year-olds in the United States with alcohol use disorders. That means they're binge drinking on a regular basis, and only a very small percentage of those are actually ever treated for it. So they're actually kind of hidden from the treatment environment, and yet they're out there. And they're the ones, of course, that you as educators have to deal with because they have really severe educational problems. This is a slide that I actually, from Susan Tappert's lab, that was actually Bonnie's mentor at the University of San Diego. Isn't that, yeah. And basically what they're looking at here, here's a normal 15-year-old, here's a 15-year-old that was a heavy drinker. And what they're asking these people to do in the MRI environment is to learn a task. And when you're learning a task, the brain is activated, and you can see the nice activation here in the non-drinker, and you can see the consequences of what happens in that same task in the heavy drinker. So in the 15-year-old, we have the same data in adults. And so there are giant consequences here, and for learning and memory. I think that's another thing we want to stress as we go through the night, in these young individuals that are abusing drugs. This is a tricky little thing here. This is just some data that I think you're all aware of. We're going to go through it very quickly. We're looking at drug use in the past month in the 8th, 10th, and 12th grade. And it turns out even in the 8th grade, we see that about 15% have used alcohol at least once. By the time we get to the 12th grade, more than 40% have. Here we can see this is cigarettes, this is marijuana. Marijuana, of course, being the most widely illegally used substance in any age group. But certainly in this teen and preteen group, we see a heavy marijuana use. And by the 12th grade, about 20% of them are using marijuana at least once in the previous month. These data are from 2007. Here's some data from a recently published study from the University of Michigan, looking over time now at marijuana and cigarette use, beginning in '75 up to the current period. We can see that actually cigarette use and marijuana use to some extent have actually declined, but we're now at a pretty stable period where both cigarette smoking and use of marijuana is used in about 20% of the 12th graders in the United States. This is an interesting one. So about 6% of, so over here you want to follow the daily use one, about 6% of 12th graders use marijuana daily. So if you have a large high school with 500 kids and students in the 12th grade, I guess it would be a very large high school, at least 30 of them then, on average, would be using marijuana on a daily basis. If you ask children in the 12th grade, I guess not children, young adults, excuse me, when you ask them, do you think that's a risk to be smoking marijuana every day? Only half of them think it's a risk. So there's a half that thinks it's not a risk. And they think this is okay. And so somehow that message needs to get through that this is not okay. And that's one of the things, again, I think that we need to talk about tonight. This is a summary of what's being abused by the 12th graders. And the drug that's being abused most, of course, is marijuana and hashish. You'll notice if you go along here, and about that kind of accounts for about almost 35% of drugs that are abused. If you go across the list here, what you're going to find is that a lot of the drugs that are abused are things they picked out of a medicine cabinet somewhere. Vicodin, for example, cough medicine, Adderall, tranquilizers, Oxycontin, for example. Interestingly, the one here that actually has a fairly moderate use is salvia. Of course, this is still legal in Oregon. You can buy this at health food stores or online. Salvia, those of you who have a pharmacological bent, is both a kappa opiate receptor agonist and a partial agonist at the D2 receptor, things that we know affect behavior. And it produces, if you take enough of it and you get the pipe hot enough, it produces a kind of a hallucinogenic trance, which has a very long life. In the West, I think California is the only state which bans the use of salvia. I'm pretty sure that's actually correct. Okay. I'm going to go back a slide. I showed this slide last, I guess it was on the 21st, on that Monday presentation. I love this poster. This is a real poster from right during the beginning of the Second World War, where we actually encouraged our pilots to take amphetamines because they would be better pilots than the Canadian pilots. I'm not sure, I didn't know what the competition was here, but for those of you that actually know or have witnessed the effects of chronic amphetamine abuse, it became clear very soon during the war, not only for the United States, but in Germany and in Japan, that taking amphetamines on a regular basis led to something like this, this kind of psychosis that we know that is very common. And indeed, the first report of amphetamine-induced psychosis that we know about, at least in the literature, was in 1938. I mention this because what I did for my PhD dissertation was look at the effects of very large doses of amphetamine in animal models. I was very interested in how this affects things. We know how it affects neurotransmitters. I was very interested in how it produced long-term effects and other systems in the brain. And perhaps and I'm not going to show you any data on this, but this I think is important. Okay, what we found is that this is just a cartoon of protein synthesis, and this is a cartoon of lipid synthesis in the brain. And of course, these two combine to make membranes and all the proteins that make us function. What we found is that in the animals that had chronically abused, I chronically abused them, I have to admit, amphetamine for a long time, we took the drug away, there was a long-term persistent decrease in brain protein and lipid synthesis. With that kind of decrease, I can tell you these animals couldn't learn anything. And so we have to suspect that in individuals that are abusing amphetamines to that same extent, that we're going to see the same sort of decrements in these the fundamental machinery of the brain and perhaps the whole body as well. But certainly the effect on the brain is terrific. Okay, beginning about in 1988, I had the good fortune of working with Nora Volkoff, who you're going to hear, I guess it's about 2 months from now. Is that right? Yeah. And Nora and I had an interaction and a collaboration that lasted almost 20 years. And what we were looking at, and this is kind of a follow-up to my earlier interest in neuropathology and the neurotoxicology of drug abuse, we were very interested in what are the long-term effects on brain processes and individuals that have abused drugs. We primarily focused on alcohol and cocaine, but we also did a bit of work on heroin as well. And the technique that we used to look inside the brain is positron emission tomography. This is a radioactive technique. Positrons are positively charged electrons. You label your compound of interest with a positron emitting ion. It's injected, it gets in the brain, and the positron comes in contact with an electron. There's annihilation, light streams out of the brain. And there's a ring of detectors. Have you ever seen a PET scanner? And it simply picks up the light, and then the computer reconstructs an image. Some of you know probably about the dopamine system and its critical role in the brain's reward system. And of course, most drugs of abuse hijack the system and use it to produce the euphoria, which is the pleasant part, I guess, if you will, of abusing drugs. We can image these dopamine receptors because they have such a high concentration in the brain. Here we're looking at the dopamine transporters. These are these things right here that take the dopamine out of the synaptic cleft, and they terminate dopamine's action. We can look at the dopamine receptors. The picture looks almost exactly the same because one's in the front, one's in the back, if you will. We can also look at the brain in another way. We can look at brain metabolism in general. Here we use a variant of glucose. Glucose, of course, is what we need to produce ATP, which is the brain's main energy source. And so we can look at glucose metabolism. And here we're looking at the enzymes, especially monoamine oxidase B, which is involved in the metabolism of dopamine. And that'll become interesting a little bit later on. So this is a summary slide that I've used for years, but I can't find a better one. And this is really, I think, part of the problem we have when we're dealing with drug abuse and how we're going to actually try to fix the problem. Here we have an individual. I actually happen to be the normal brain right here. Here's the addicted individual. This person was abusing cocaine. This person was abusing alcohol. This person was abusing heroin. All those drugs, one either directly or indirectly, end up releasing dopamine or causing more dopamine to be in the synaptic cleft. The receptors that are out there respond in exactly the way you would predict. They downregulate. They say, aha, there's too much here. I have to accommodate this. The machinery is organized, and all of a sudden what you have, the receptors are either down or downregulated. And so the consequence of that in the reward circuits, here's the normal reward circuit in the non-drug abuser. Here it is in the drug abuser. And what you can see now, of course, is that the number of these, the reward circuit is downregulated, and so of course then what does the drug user do? They start taking more drugs. So the brain's doing exactly what it was trained to do from an evolutionary perspective, but the consequence of that is it leads to increased drug use. The interesting part about this downregulation is here. This is a cocaine addict. This is a normal subject up here. Here's the cocaine abuser. This is 1 month post-cocaine use. And this is 4 months post-cocaine use. Now, some of you may ask, how do I know these people weren't using cocaine during this period? This particular study we did in 1990, we could actually put them on a locked ward in the VA. And we actually tested them 3 times a week. Because believe me, you get on a locked ward, drugs get in. These people had not used drugs. And what we can see here, here's normal. Here you can see the marked down regulation of these receptors. Here you can see even 4 months later that they're still abnormal. I'm not going to be able to explain this all to you today, but we have good reasons to believe that this downregulation that we see, this difference between the drug user and the normal, is both due to the effects of the drug and to the fact they probably had lower receptors to begin with. So a risk factor, a genetic risk factor, if you will, is lower dopamine receptors. And we could talk more about that in the discussion period. The most dramatic picture I want to show you, perhaps, is this one. We're now looking at brain glucose metabolism in a cocaine abuser. Here is a normal subject for different slices of the brain. You can see the nice activity around the cortical rim. Here's a cocaine abuser, 10 days post cocaine withdrawal, and here's 100 days post withdrawal. If you showed me these pictures and didn't tell me they were cocaine abusers, my initial guess would be this is somebody with Alzheimer's disease. Okay. So the point I want to get through, and if I can get no other point through today at all, is that whether you're 15 or 25 or 35, once you stop taking drugs, the brain takes a long, long time to get well. It's been injured. This is different than the kind of the psychological things we think about of drug abuse. Those are all important as well. The point is the brain is injured. Just think about it like I hit you over the head with a baseball bat. If that happened, you'd go to the hospital and you would be ready for therapy for months and months to recover from that injury. I can think of House Representative Giffords, who was shot in the head. Her recuperation is going to take months, if not years, to get over that kind of injury. The heavy drug abusers are injuring their brain, not the same way, of course, obviously, but the injury is there nonetheless, and the recovery period takes time. So simply getting kids to stop taking drugs doesn't solve the problem. There's a real underlying physical problem that's there, and it's going to stay there for a very long period of time. Here, this is one thing that you notice, and you probably have all noticed, that people that, again, teens that abuse drugs, they also smoke a lot. It's almost impossible to find a cocaine user or a heroin user or a teen alcoholic who doesn't smoke a pack of cigarettes a day. We actually figured out in part why that may interact together. I'm not going to tell you the reason why we were looking at a ligand, or a PET ligand, positive emission ligand, to look at monoamine oxidase B. Again, this is the enzyme that metabolizes dopamine. This is a non-smoker, and here's a smoker. And it turns out there's something in cigarette smoke that inhibits this enzyme that's critical for regulating the concentration of dopamine in the synapse. It's not nicotine. Okay? It's a flavonoid that's in the tobacco plant. It's not something the cigarette companies put in there. It's just in the plant. Okay? And there are several different flavonoids that produce this effect. But the consequence of having something like this there is illustrated in the next slide, which is here. So here we have someone that's smoking, getting their dose of nicotine, and they're also drinking at the same time. Nicotine releases dopamine, alcohol indirectly releases dopamine, and so it gets released. Normally, to some degree, monoamine oxidase B is around to kind of sponge up all this extra dopamine that's out there and kind of regulate what's going on. But if you're smoking, okay, it's not there. And so you actually get a bigger bang for the buck. So smoking, not only through the nicotine, but through these other compounds that are in the cigarette smoke, actually potentiates the effects of drugs of abuse. And so it becomes much more easily understandable now how this interaction between smoking and other drugs of abuse has come about. I'm doing it the wrong way. I often get asked at talks like this, what about Ritalin? Ritalin, of course, works almost in exactly the same way, or not exactly, but close enough, as cocaine. And the question is, why is it that we can give Ritalin to kids with attention deficit hyperactivity disorder, and why don't they become addicted and whatnot? It turns out that there's a good reason for why that occurs, and that's really illustrated here, and I'll just summarize very quickly, is that if you take IV cocaine, the drug gets up in the brain very quickly. If you take IV Ritalin, the drug gets up in the brain very quickly. Cocaine addicts cannot tell the difference between these two. So if you take a cocaine addict and give them IV Ritalin, they claim it's cocaine. Notice no difference whatsoever. If, however, you give the Ritalin orally, it actually gets up into the brain at about the same occupancy of the dopamine transporter, but it gets there very slowly. And so the reason why you feel, it appears, in part at least, the reason why you have the euphoric response from cocaine and IV Ritalin and you don't get it with oral Ritalin is the rate of uptake into the brain. Although at the end, the occupancy of the drug in the brain is about the same. So it's that rapid uptake into the brain which makes it a drug of abuse. Now we're going to switch to Oregon's favorite drug, methamphetamine. Methamphetamine, like cocaine and Ritalin, works on the dopamine transporter, puts more dopamine in the synaptic cleft, and that's presumably the main part of its mechanism of action. We did a study, again, remember that I'm doing this study in Long Island at Brookhaven National Laboratory. We couldn't find any methamphetamine addicts to participate in this study in New York. And the reason is interesting because the mafia simply would not allow methamphetamine to be sold. They had a market on cocaine. You're going to take cocaine, you're not going to take methamphetamine. And really the only amphetamine addicts that we could find in New York, and they didn't usually use it very much, were found in the gay community. So the subjects in this study were actually flown to Long Island from Los Angeles, where there were lots of methamphetamine addicts. And they all were participating by judge decree in an abstinence program. Most of the participants in this study were women, and if they didn't stay abstinent, they were going to lose their children. And so it was kind of a, if you will, a contrived sort of study, but from the point of view of what we learned, I think it was very important. Here is the normal brain. Now we're looking at the dopamine transporters, and we're using actually Ritalin to label those transporters. Here's a subject 80 days into detoxification. You can see there's a marked decrease in these transporters. And if you actually plot this out, and it's going to be hard to see in this one, even a year later after detoxification, they're still abnormal. So they're still not in the normal range. This we believe, and again, I don't have time to explain this, this we believe is the direct effect of the drug. So through a mechanism that's not entirely well understood, but certainly a drug-mediated mechanism, the dopamine terminals are actually depleted of all the things that make them work. The machinery goes away. And so we actually, when we measure this in the PET camera, we see this decrease. The other part of this, I forgot to put this slide in here. How do methamphetamine addicts compare with, if you will, Parkinson's disease patients? Here's a Parkinson's patient here. Again, we're looking at the dopamine transporter. Remember, Parkinson's disease is due to the degeneration of those dopamine neurons. Probably all know that. Here's the normal control. Here's a methamphetamine addict. You can see methamphetamine addict looks more like the Parkinson's patient than the control. You can make your methamphetamine badly. You end up with methamphetamine, but you still get the same effect. You just need to take more drug. If you haven't done this as an educator, Google making methamphetamine. Do this. The first thing that comes up is the bottle approach to making methamphetamine. We put all your ingredients in a bottle, a little anhydrous ammonia, take the lithium out of a battery, you take some pseudoephedrine, you put it in a little toluene, a little bit of water, and you shake it up. It makes methamphetamine. Take my word for it. This is available online. The good news in Oregon is that we have banned the sale of pseudoephedrine and ephedrine-containing drugs over the counter. That has led to a 96% reduction in the number of methamphetamine labs in Oregon. It's an impressive. The only other state that's done the same thing, and it's amazing, and you ever think this state would do it, is Mississippi. I mean, I don't have to make fun of Mississippi. Okay. I'm going to go fast. This is the last. This is on these pictures. This is the last one of these. Methamphetamine does something very different than the other drugs of abuse, and this is important. Methamphetamine causes an inflammation in the brain, which we saw on the PET camera initially as an increase in glucose utilization. But what really was happening here was a reactive gliosis. The glial cells are increasing in number, and neurons are dying. Amphetamine does the same thing. Cocaine doesn't do this, heroin doesn't do this, alcohol doesn't do this. So this is a very different picture. But again, it's obviously grossly abnormal. You can see the control, you can see the methamphetamine addict. This picture was taken 90 days into withdrawal. So the brain is still very unusual. These studies have actually been also looked at in monkeys, and this reactive gliosis can last for more than a year and a half after discontinuing the drug. So this is a very long-term effect of these drugs of abuse. Again, when you're dealing with things like, I want to teach them something, learning and memory, the brain is simply not able to function at any really reasonable level. I want to turn now just real quickly, and I'll be done in 2 minutes, I think, with some genetic studies. Drug abuse is highly heritable, and it's heritable in a way that compares to other disorders. Here we see, for example, this one? Nope, I'm sorry. Back. Good. Okay. Most are probably familiar with these data. Something like bipolar disorder has about, these are twin studies, has about 80% heritability among twins. So there's a very small environmental effect there. Schizophrenia is about the same. Alzheimer's disease is obviously highly heritable, you know about that. Panic disorder, depression, generalized anxiety disorder, somewhat less so. Here's alcoholism. So the heritability among twins is a little bit more than 50%. I've seen other data that has this a bit higher, some a bit lower. The point is that almost all drugs of abuse, there's a genetic component there. And we can see this very nicely in twin studies. And many of the twin studies have been done where the twins were adopted out at birth, living in an environment different than the home environment, where there's likely to be a lot of drug abuse. And you still see the same, basically the same data. So there are risk factors there that are predisposing them to abuse alcohol and other drugs of abuse. Do we know what those risk factors are? Not exactly yet, but we're all trying to look at those. And so up here in your top left, here we have the brain and the DNA. So we want to know what this brain-behavior relationship is. Luckily, we have new tools at our hand that are making this possible at a rate that even 3 or 4 years ago, I would have thought absolutely impossible. This little machine right here is about the size of 4 microwave ovens. This is a next generation sequencer. This is the machine that can sequence your genome in less than a week. This right now, that cost is about $10,000. By the end of the year, it will probably be about $2,000. And in 3 years from now, the cost will be down to about $100. So we're going to actually have genomic information. It's probably likely to become part of routine medical care to know what your genome is. How that information gets used and used well or used badly, of course, is a great ethical concern, and we can talk about that if you'd like. The point is that we can now take individuals, or we can take complex animal mixes and animal models and things like that, and we can actually sequence the genome very quickly, and we can try to then figure out what genes are associated with that behavior. We now have new tools at our hand that allow us to do this. And this is really a really fascinating and exciting part of what's going on in medicine. And the last slide, you gotta do it again. Oh, it's one of these. I borrowed this from Tom Insel, who's the director of the National Institute of Mental Health, and I'm just going to real quickly just put it all together here for you. The point is that these new technologies are going to lead us now. We're going to learn something about what genes are involved. We'll learn something about what cells those genes are associated with particular cells. Those cells are associated with particular systems, and then as we go through from the gene all the way up to the systems to the whole individual, what's going to happen with the new trends in medicine is it will end up here somewhere with prevention and cure therapeutics and personalized care, so that we'll actually be able to know which individuals are at risk for developing these disorders, and we can prospectively then begin interventions. So we don't wait till we actually see them in the clinic, where by this time they've already done a considerable amount of damage. And so we're very close to this sort of strategy is used every day in cancer treatment. We have not adapted this so much yet to the treatment of mental health disorders or drug abuse disorders, alcohol abuse disorders, but it's coming. And so we'll actually then be able to really, I think, improve the strategies that we use to intervene in individuals. So I think there's only one more slide, and I've almost got it at the right time. Again, coming back to this slide, is that what you're interested in as educators is right here in the middle. Their development, how they're working, their social and emotional competence, their brain maturation, puberty, their ability to learn in the classroom. Drugs of abuse will affect this through affecting physiology. Genetics have a role in this. Behavior has a role in this, and of course the environment has a role in this. So without a comprehensive picture of all these things that are going on and having that integrated well, it's probably unlikely that we can solve the problem that we have before us. But I think if we think of it standing, if you will, a bit at 10,000 feet and looking at all these intervening variables, that we can be very successful. Thank you.
So we're going to move to the panel seating location over here. I'm going to take all the technology with us, so we're ready to proceed there. Portland to join the Primary Center and OHSU. She served as director of a multi-university consortium on stress, anxiety, and alcoholism since 2001, and is currently serving on the National Advisory Council to NIAAA. So welcome, Dr. Kathy Grant.
Thank you. Is this on? I've not really given a talk seated before. So you can't shout out, sit down. I'm already sitting. Anyway, okay, so Bob had trouble with 2, and now I'm going to have 3. Okay, so my comments are directed more towards you as educators and giving you a follow-up on Bob's talk, some things to think about in terms of what we're finding out in our animal models that might help you understand if you have children in the classroom who you think are affected and or who are on a trajectory towards trying to learn under the influence. Oops, boy, you're right, this is tricky. Okay, so I'm going to start out with this slide because it has the age of relevance for many of you, and that is the young teen. Pointing it to myself. And there's 2 things here, and one is if you come from a family history of alcoholism, then those individuals are in blue. And if you come from a family history negative for alcohol, there is no alcoholism in your family, then that's plotted in red here. And the point is we can take the most at-risk children, the ones coming from families where there is alcoholism, and you can see that the 13, if they start drinking when they're 13 years old, the chances in their lifetime of becoming diagnosed with alcohol abuse or alcoholism is 4-fold higher than if they don't start drinking until they're in their 20s. This slide right here, and even if this is largely a genetic risk, although the family environment growing up in an alcoholic household is beyond genetics, even if you took that away, you can see that if you start drinking when you're young, you have a lifetime risk. You've put yourself at a different risk than if you don't start drinking when you're young. The question here, though, and why animals are important in this research is if somehow miraculously, because we have laws, right? We try to keep everyone protected from drinking until they're 21. But of course, alcohol is this really interesting compound. It's only 2 carbons with a single oxygen. It goes to every cell in the body. It can affect every organ. And when it gets, it's almost ubiquitous in our society. So not as only ubiquitous in our body once we drink it, but it's ubiquitous in our society. If we could keep children away from alcohol, would they still have that same course, right? So is there something genetically predetermined in these children, or in fact, is it a critical window? And if you could keep them from drinking through that critical window, would they no longer be at risk for a diagnosis of alcohol abuse or alcoholism? Well, we can't randomize these children to these trials. We're already trying to keep them away from alcohol. The only way really to sort this out, because humans can be self-selecting in how they use their time and what drugs they come in contact with, we can randomize animals, young animals, and older animals to studies like this. And we can then determine, is there really a biological risk window, vulnerability? If we could get them through this vulnerability, they would in fact be much healthier individuals in our society. So that's one of the first points to make. Oops. Man. I'm really sorry.
It takes practice.
Yeah.
Do you want me to do that for you? Just say next slide? Is that
No, that's okay. If I give anyone a seizure, I'm very sorry. So to ask these questions, we asked them in macaque monkeys, in particular because their adolescent phase, these are our old world cousins, and their adolescent phase is about 1.5 years long. So it's a long enough period of time where if we then gave them access to alcohol and trained them to drink it, we could ask them later in life, or continue to drink, how much would they drink. And plus, our old world cousins are kind of like us in terms of the percentage of animals that will go on to be heavy drinkers. And the way our brains are probably related to our behavior is much more involved in cognitive abilities and cortical development. And so it's rare for an organism like a rodent to find intoxication something that they want to repeat. But it's not rare for primates to find an altered state something that they find that they would want to repeat. So in terms of what animals you want to use, there's some face validity in looking at least at heavy drinking with the non-human primate. Oops. And we have, I'll just mention really quickly, we have a lot of tours out at the Primate Center, and we encourage you to come out, and especially educators. We have lots of school tours as well. So what are we finding in the monkeys? Well, we're finding that if they start drinking, so this is kind of a complicated slide, but really we started animals as young adults or as adults. These young adult animals are probably around the age of 17 to 19-year-old equivalent for us, and the adults were probably about 25 to 40-year-olds. So not many of us in the United States anyway have our first drink after 25 to 40-year-olds. But there are emerging economies in the world in which this is happening now. And then there are these young adults. And what we found is the percentage of days, and this is over a 1 year, 365 days, the percentage of days where animals drank alcohol, what we would call heavy drinking, which is over 4 grams per kilogram. This is a 16 drink equivalent. So over 16 drinks a day. If they didn't start drinking when they were adults, very rarely did they have this heavy of a drinking day over the year. And you can see that the animals that were started as young adults, though, this guy up here, more than half his days did he drink over 16 drink equivalent per day. So we think we're getting very close to really answering the question of, is there a vulnerable period? And can we get people through this risky period and have a much healthier population as a result of it? And let's make sure, okay. So then the other thing about animal models, and this is largely not coming from primate research, although some of my research is addressing this, this is largely coming from rodent research because in these studies they're not necessarily looking at drinking per se, but they're looking at the effects of alcohol. And we know that if we give adolescent animals compared to adult animals a history of alcohol, so we actually administer the alcohol, we know that these adolescent animals are less sensitive to the sedation effects of alcohol. They don't become sleepy or tired or drowsy as easily. They're less motor impaired. They don't have as big of a hypothermia. They don't have as great of an analgesia. They're not as socially inhibited. And they have a much less severe withdrawal or hangover. So adolescents, when they drink alcohol, are not as sensitive to these effects. But they have a greater sensitivity to an impairment in an area in the brain called the hippocampus, which is really important in laying down our learning and memory. That it impairs their hippocampal. This is called long-term potentiation. It's our model of memory in rodents. And then if you look at their behavior, they're impaired in terms of spatial memory in what we call a water maze, but many other mazes too. So it's as if they don't really remember that they explored that area and they go and explore it again. And there's a facilitation of social behavior, which matches well with a lower, not as inhibited and a greater social behavior. So this is really what we're coming from the animal literature some really important targets, if you will, to understand how, if children are using alcohol, how it might be affecting their behavior within the classroom. This is inside the body. This is a chemical pathway. This is all happens inside us, where we can take cholesterol either in the adrenals or the brain or the liver. It goes through a couple steps to progesterone. And why am I showing you this complicated pathway? It's because these byproducts, or these products of these different enzymes, which are all gene products, by the way, so you could have different genes for this particular enzyme right here. These go down into these steroids. And these steroids are, I've labeled them as being either GABA active or NMDA active. These are neurotransmitter systems in the brain. They're the largest neurotransmitter system in our brain that controls our excitatory function, what keeps us awake, and our inhibitory function, which helps us sleep. Also, the balance between being inhibited and excited is really important for effective learning and memory and understanding and attention and keeping to a task. So when something endogenously, like a progesterone with menstrual cycle that's coming in during adolescence, and I don't show it here, but this pathway here up to androsterone, you can get there through testosterone, which is also coming in at adolescence. But the byproducts of these are these brain actives. So the body does go up to the brain through our hormones and interferes or interacts at least with these brain. So puberty is a really interesting time, and it is a moving target. So it's really very hard to understand what's going on in puberty without animal models. And we know much of what's going on in puberty with animal models. And then I think I have one more slide. This is my final slide. The other one's just in case questions come up. It's just a summary slide of what we know right now. What we know is that in male rhesus monkeys, there is an effect of early versus later adult onset, and now we've added a younger group targeting the equivalent of a 14-year-old, 14 to 16-year-old in that study. This is very important that we're just coming out with both the behavior and the changes in the brain. Heavy drinking in an adolescent animal disrupts attention, learning, and memory. Now, we know this from rodent studies. We're learning this also, establishing this in monkeys. But what we do know in our monkeys is more likely with heavy drinking that the animal is going to engage in habitual behaviors and less sensitive to prefrontal cortical input. What that means is if a kid has a habit, or somebody who's young and drinking or even older and drinking, they're more likely to use habitual behavior. They're much less likely to have cognitive input coming down from the cortex to change that behavior. It becomes very hard to change ingrained behaviors in a heavy drinking individual. Social behavior is affected. Animals become less inhibited and more likely to be aggressive. Very heavy drinking can be associated with signs of depression and other comorbid disorders, and this is very clear also from the human literature, and we're very interested in the mechanisms of that. And then finally, stress and stress hormones, which all those adrenal hormones essentially are stress hormones, are elevated either directly or with subsequent outcomes, because kids aren't supposed to be drinking. They know they're not supposed to be drinking. That's an increase in your stress system. Even if they're going to say it's fun and exciting and all that, the HPA axis is on with these kids. They have to deal with that stress. They especially have to deal with that stress if something bad happens. So the way stress is handled in the body is really altered by a baseline of heavy drinking. So subsequent outcomes can alter the capacity to be calm and also to be non-impulsive. We know that individuals that are really highly stressed and animals that are highly stressed are likely to do impulsive things. And they're also likely to do, once they do the impulsive thing, and it sounds counterintuitive, then they're likely to go on to a habitual behavior. So they do something rash, and then they go and they do a stereotype of something that they're used to doing. With a heavy drinker, what that means is they're going to do something rash, and then they're going to go and drink heavily. And I'll stop there. Thank you.
Our next panelist, Dr. Bonnie Nagel, earned her doctoral degree in clinical psychology with an emphasis in neuropsychology from the University of Memphis. After completing a 2-year postdoctoral fellowship and a 1-year academic appointment at the University of California in San Diego, what slides you saw earlier in Bob's presentation from that lab, Dr. Nagel then joined the faculty at OHSU as an assistant professor in the departments of psychiatry and behavioral neuroscience. That was in 2006, pretty recent. Welcome. So great. Dr. Nagel's research is focused on understanding adolescent neurodevelopment, individual developmental differences, the development of executive functioning, some of which we were just talking about a moment ago, and how variations in and interactions among these variables are associated with the emergence of psychopathology, particularly alcohol use disorders. So the next panelist is Dr. Bonnie Nagel.
Thank you. Okay, so I think we heard a bit from Dr. Hicksman about kind of acute and sustained influences of drugs on the adult brain. And Dr. Graham talked a little bit about adolescence and what alcohol does to the adolescent brain and why adolescence might be a period of vulnerability. I'm going to shift gears a little bit and talk really about the human adolescent brain, what's developing, why it is a period of vulnerability, at least to the best of our knowledge, and what then does alcohol in particular do to the developing vulnerable adolescent brain. I'm a little nervous about this because I have a lot of transitions, so we'll see how it goes. It could be very frustrating. Okay, so starting with adolescence, certainly there are a lot of positive changes that are occurring, so we don't want to paint it entirely as a negative time in life. There are lots of improvements that are occurring. And many of those are in the realm of cognition. So there are lots of improvements in cognitive functioning that occur across the adolescent age range. And those include, but are not limited to, the ability to multitask. So the ability to take on multiple demands at one time and really simultaneously conquer and effectively do those different tasks. I think this plays important relevance for many of the demands that we place on adolescents, both in school and in society. We can think about some of the things that have been in popular media about driving, the adolescent brain, and what it's like at 16 to really be driving and dual tasking. And then you add on complexity of what is it going to be to the adolescent driver who's also talking to their friend in the car or trying to text, etc. So it plays real relevance that this is a function that's really improving across the adolescent years. The emergence of cognitive and emotional control and the ability to inhibit and regulate these functions is coming on during the adolescent years. And although it's improving, it really isn't something that happens overnight, and it's something that takes a protracted course of development to really reach maturation. We also see across adolescence an enhanced ability to plan and appreciate long-term consequences. Certainly this plays a role when we're talking about the influence of alcohol and drugs on the adolescent brain, as many of the consequences that we're describing don't occur with one incident of use and really are more long-term and perhaps sustained in their effect. We also see increases in social awareness and social importance, the importance of peers during the adolescent age range, and as well as the ability to do perspective taking. So all of these really important cognitive changes are occurring that I think certainly in all of your experience make the 12-year-old very different in most cases from the 18-year-old. There are a number of different things happening at a brain level that are supporting these changes. We see that although the brain size is largely complete and largely stable after about the age of 6, there are a number of different progressive and regressive changes that are occurring. And in particular, we see the white matter of the brain. This is the axons of the brain, the part of the brain that allows neurons to communicate with one another. This is getting bigger, and it's getting bigger because likely due to myelination, which is a process that speeds neural transmission. It allows, this is where it becomes challenging. it allows this region of the brain to more quickly communicate with this region of the brain. That's important when we're talking about cognitive functioning, the ability to make decisions quickly and in different types of situations. In addition to the increase in white matter and the myelination and speeding of neural transmission, we also see that gray matter gets smaller. And essentially what this is a pruning back of spurious connections that are no longer being used and are no longer necessary for the brain to process efficiently. Importantly, the regions here highlighted in green are the frontal lobes of the brain, and this is the area of the brain that develops the most during the adolescent time period. And it is part of the brain that we think subserves most of the cognitive functions that I described as improving across these years. When we look at functional tasks of the adolescent brain, we see that in many development areas, including the prefrontal cortex that I have highlighted here, adolescents have to utilize that part of the brain and work harder to complete task demands at the same level as an adult. Although they may be able to do so, they may be able to problem solve, they may be able to attend at the same performance level as an adult, they're working a lot harder at a neural standpoint to do so. So that's sort of the positive. I think it's important to start with that. But as we all know, in working with adolescents, this is a time of vulnerability, and Dr. Grant highlighted that nicely. Why? Why is this such a period of vulnerability? I think importantly, one of the things that we know about and we certainly see every day that we interact with an adolescent is their emotions. And this is a very important thing to talk about. Adolescence is a time of increased emotional intensity and responsiveness. And again, all of you who have worked with adolescents or have raised adolescents or have even been an adolescent know this. And it isn't something that's trivial, because in fact, an area of the brain that's associated with the perception and processing of emotion is the amygdala. And when we look at the way that the amygdala responds in adolescents compared to adults, we see that when presented with negative emotional stimuli, the amygdala in the adolescent is much more active than in an adult brain. Now this parallels very well with both subjective and more physical reports of stress, whether it's galvanic skin response, kind of a stress test, to different types of emotional stimuli, or if you were to ask an adolescent compared to an adult, how stressful was this negative event for you? Adolescents are always going to rate this, or by and large going to rate this as more stressful than any other period of development. And I think that plays an important role in understanding adolescents and understanding how their brain is processing information. Interestingly, and Kathy sort of talked about this too, there has been some evidence that some of the gonadal hormones or the pubertal hormones that are changing obviously during the adolescent years are related to this emotional responsiveness. And so thinking about when puberty is occurring in males and females, it's certainly much younger than it used to be. And we can think about it as 9, 10, 11 years of age when these pubertal hormones are really becoming activated and this emotional responsiveness really takes over. The other thing that we see in adolescence that I think confers vulnerability is a kind of a difference in motivation and a heightened risk-taking. Now this is certainly not true for all adolescents, but by and large, if we were to look at adolescents compared to adults, when they are presented with a risk-taking task, and here the choice is a kind of a 30-70 odds, so it's less than 50-50 chance that you're going to win. It's a little risky. It's not the most risky that you could possibly have, but it definitely is riskier than, say, 70-30. What we can see here is that the 10-year-olds are making those types of choices about 75% of the time. Whereas if we follow these people up into their 30s, you're going to see that it comes down to less than 50% of the time that an individual is going to take those odds. Some of the work in our own lab parallels that nicely and suggests that, yes, adolescents, not just the sample, but adolescents in general, do make more risky decisions. So I talked about the good in terms of all these positive cognitive changes that are happening, better cognitive problem solving, emotional regulation, and I also talked about sort of the awakening of emotion and the awakening of risk-taking and changes in motivation that occur in adolescence. So how does the development of these two systems respectively result in this vulnerability that occurs during adolescence? Well, we have this reward and emotion, this motivation system that I talked about second. We can call it the bad. We'll get to the ugly later. This really happens early in adolescence, and if we think about it as being associated with some of the hormonal changes that occur, it's happening very, very early. We have the prefrontal cortex up here in the front of the brain that is responsible and is developing across adolescence. And as Dr. Hitzman showed, there was if you remember the bright colored slide that I'm sure many of you have seen many times, the prefrontal cortex develops all through adolescence. And so we really see again a protracted course of development that subserves many of these higher level cognitive and regulatory functions of the brain. Importantly, the connections between these two regions are also immature during adolescence, and that there confers the vulnerability. You have sort of the awakening and the heightening of appetitive and reward and emotional systems in the brain well before regulatory systems are either mature and or connected to come in and regulate some of these functions. And that makes adolescence a very unique time in life that allows them to be vulnerable to developing things such as addiction. I have far too many remotes in my hands. All right, so adolescents in general tend to use emotion and reward areas of their brain to govern behavior as opposed to some of these more elaborate frontal systems that hopefully many adults use. Certainly there's a difference between hot and cold situations, and we can talk about that later, but if you ask an adolescent at the dinner table, with parents or with your teachers, sitting in the classroom in a cool situation, what's right and wrong, what do you do in these situations, they're able to tell you. It's not that they're ignorant. I'm not by any means suggesting that. But if you place them into a heated situation, what happens is these reward and emotion systems really override. They're more active, and the frontal systems that regulate them can't come in fast enough to govern behavior. And this is the increase for developing psychopathology. So this is the same slide, maybe a little bit more recent one that Dr. Hitzman showed. The importance here, and I'm going to highlight alcohol use as one of the substances, although we know that marijuana use is quite high as well. We can see again, just to remind you, that if we look at past month use, we've got about almost 50% of 12th graders using alcohol, and a little over 15% of 8th graders. And then when we look at past month drunkenness, so drinking so much that one experiences feelings of being drunk, we have 12th graders showing over 30% in the past month have drank to that level. So certainly it's a large problem. When we look at the effects of alcohol on the teen brain, I think this is something that both of our previous speakers have alluded to, and it is really important. What we can see here is that when we look at alcohol-dependent teens in treatment, we see that for both verbal and nonverbal information, the alcohol-dependent and non-using, they're not using at this time, teens, retain about 10% less information than their non-using counterparts. Now that may not seem like a big deal. It's the difference between an A and a B, or it's the difference between a passing and a failing grade. 10% of the information that they have gotten in, they're not retaining. So assuming that they actually are getting it all in. Importantly, when we look at brain structure, we can see abnormal prefrontal cortex development in heavy alcohol-using teens, and then that is the region that's developing during this time frame and is responsible for the development of those regulatory functions. Oops, there, I knew I was going to get one of those this time. We also see reduced hippocampal volume. This is again, as I think one of our previous speakers alluded to, the part of the brain that's necessary for encoding new information and learning new information. And we see that the volume of that structure is reduced in heavy binge drinking adolescents. These are youth that are not in treatment, but just do your binge drinking that we see on a regular basis in high school students. This picture is a little bit different than the one that Dr. Hitzman showed, and essentially shows you a 16-year-old non-drinker and heavy drinker. And what you can see here is with comparable task performance, the 16-year-old heavy drinker is utilizing a lot more of their brain, this is a functional activity study, in order to perform at the same level. When you get to age 20, though, and 5 more years of drinking, you see that the non-drinker is using an adequate amount of their brain to perform a task, And at this point, the person who's been drinking for 5 years is no longer able to compensate. Their performance is actually falling down, and you see that paralleled by lack of activity in their brain. So essentially, importantly, the adolescent brain is still developing. I think that's not news to any of us here. But due to the timing of the different systems, the early maturation of reward and systems that are implicated in emotion and the addictive process, and the later developing regulatory systems, adolescents are particularly vulnerable. Substance use impairs cognition in these youth. It's worse with heavier use. It's worse with withdrawal symptoms, so hangovers. And of course, the longer you use, the worse it's going to be. And paralleling their behavior, we certainly see a number of different structural and functional abnormalities. Again, it may seem like a modest severity, 10% difference in their memory retention, but this has a broad influence. And this is something that, as educators, all of us should be very concerned about.
Thank you, Mona. All right, we're going to pass off this multiple techno wizardry to the one homie in the crowd here, Ellen, who's from the University of Portland, who hopefully has used this lecture hall and this apparatus before. Let me give you a brief background on Dr. Ellen Arwood. She's a speech and language pathologist who's been working in higher education for 39 years now. She's author of 7 textbooks and numerous articles and monographs on language and learning. She's established a strong reputation as an expert in how language is used for learning and how language and cognition interact. Her development of assessment methods and instructional strategies for children and adults has positively influenced the lives of thousands of individuals, both nationally and internationally. Currently, she's a professor here at the School of Education at the University of Portland, where she teaches courses in language and neuroeducation. She's the author of the Neurosemantic Language Learning Theory, a 4-stage model for explaining relationships between pattern learning and conceptual learning, mediated through the acquisition of language. Dr. Ellen Arwood.
Thank you. Thank you. I've had the opportunity to listen to Dr. Hitzman and Dr. Grant and also Dr. Nagel. It's been really a pleasure of mine to be able to hear them again. The one thing that stands out in my mind is their insight about how we as educators can take some of their neuroscience and do something with that. So my role tonight is to try to take your neuroscience and actually do some neuroeducation suggestions. And when I think, oh boy.
I've got you headphones too.
Yeah. We'll go back.
Can you get it back?
Yeah, I held it down too long. We're going. Maybe.
Come back or forward?
I'm trying to go back. There we go.
Who's there? Okay.
Ah. Thank you. There we go. Can't hold it down. It moves way too fast. Okay, there are 3 basic areas I'm going to talk about. Because as an educator practitioner, what I think of when I think about drug and alcohol abuse is I think about prevention, intervention, and learning. And in the area of prevention, we have a lot of programs out there that are designed in the educational field to use the community, to use the educators, to use the people from the neuroscientists, the medical folks, the allied health folks, to actually look at different types of ways to prevent and deter our students from actually using alcohol and or drugs, other substances. The interesting thing about the prevention programs is that most of them don't really look at how they present the material to the students. They look at the developmental level of the material being appropriate at a grade level, but they don't necessarily use the neuroscience to actually look at how the students are learning. And there's about 33% in every classroom between pre-K and 12th grade, and I would say college, except I don't have any statistics on that, that actually those individuals function in a pre-operational, or what we call me, myself, and I type of stage, which is that the world surrounds them and they are center stage to the rest of the world. That means that nobody else is in their picture. And so when we have a prevention program and we ask them to do role plays and we ask them to look at some information and say no to drugs and we give them some statements that they're supposed to do something with, basically they memorize those, they give them back to us, but they don't understand them because they are in their own picture. And so what that says to me is that as we look at these programs, we're going to have to also start looking at their levels of development in terms of actually how they learn. And that brings us up to another piece about learning here, is that these are also my see it and do it kids. And I mean that very affectionately and respectfully, but basically if they see it, they do it. So if the pencil sharpener is there and they have a pencil that needs to be sharpened or doesn't need to be sharpened, and they see the pencil sharpener, they get up and go sharpen it. And the same thing holds true for the drugs. So when I show them how to buy drugs and I show them how to use drugs, then what I find, because I've been called in as a consultant to work with the kids, is I find that they actually can tell me which program of prevention that they went to at grade school or middle school that helped them learn how to use the drugs. Because they see it, they do it. And when they're put into those role plays, they become the buyers and the sellers and the users and the bingers and that type of thing. So it's really important that we start thinking about these students. They're easy to find. You just ask them about their language. It's my school, it's my books, I didn't do anything wrong. The teacher sent me to the counselor's office. It's not my fault. The judge sent me to the detention center. They can tell you about themselves completely, but I guarantee there's nobody else in those pictures. And so when we use prevention programs, they don't really understand those types of rules. Okay, here we go. What that means then is that for prevention, here are my suggestions from a neuroeducation standpoint, is that we need to really apply what we know about how students learn. That is how they actually acquire concepts. I'm going to talk about that a little bit more later. And I think one of the other speakers talked a little bit about the difference between habit types of learning, where you do something based upon something that you can regurgitate, and conceptual type of learning, where you actually know about what it is that you're doing, and therefore can make better choices. Our see it and do it students are able to make pictures in their head, and so when we talk, we create pictures and sometimes movies, that type of thing in their head. So when I say don't drink, there's no picture for don't. The picture they get is drink. Okay, and you know, I had a young man down in Texas that was actually expelled the first week of school. He was a football player and a 9th grader, and he was expelled because he brought marijuana to school. And they asked me to come in and do an evaluation, and I had him draw out exactly what he did. And I said, where did you get that idea, that picture in your head of how you go ride your bicycle over there and buy that marijuana? And he showed me the program and told me where he got it, how he role-played it, and how that allowed him then to buy it. His blood workup and everything, urine analysis and all, everything was clean. There were no drugs in the system, but he was expelled for a year. So that kind of brings me to the next point here. Whoops, oh, golly. I did a really good job on that one.
Want to acknowledge Hannah over here, who's the techno-fix-it person.
That's the one I want, Hannah, right there.
Thank you, Hannah.
Next one. There we go. Is intervention. Thank you. Oh, as educators, we're supposed to be in the business of education and learning. And we have a challenge as educators. And the challenge came from two folks, Dr. James Comer and Dr. Robert Pianta. Comer is the assistant dean to the medical school at Yale University and the co-founder of the Child Development Center at Yale. And Robert Pianta is the professor in education at the Curry School of Education at University of Virginia. And they wrote a booklet that basically talked about neuroscience and learning and how we really need to start using some of the material as educators. More importantly, they summarized this in the Washington Post, and they said we're not using what we know about neuroscience and learning. And I would like to accept that challenge. I basically do believe that we are not using the neuroscience. And what I mean by that is that we have to start looking at how our kids are going to be able to make choices. If in fact they have this genetic predisposition, if in fact they have an environment that doesn't provide them with the information about pro-social choices other than drinking, that type of thing, then we have to figure out how is it that they actually do learn. We also have heard quite a bit about how the drugs do change the learning system, the actual brain itself. We have to recognize that. If we have students that are on drugs and we want to intervene, then it's not a matter of telling them no, it's going to have to be, what are my other choices? What do I see myself do, and how do you draw me into my picture where I'm doing pro-social choices that are different than what I'm not supposed to be able to do, or not supposed to be doing? So it means that we have to know something about those learning systems. And we can get that from the neuroscience. We'll try this one more time. We did it. Okay. So the implications here from a neuroeducation standpoint is that if we have students that we are going to intervene because they have been on drugs or are on drugs, then we also know that the brains are either changing and or have changed. And that could be semi-permanent, it could be permanent. And we've heard a lot of data about that. I will tell you, working with some of these students, that they think and learn differently. And for us as educators to assume that we're going to use the same programs for literacy, for reading, for writing, for thinking, for viewing, for listening, for speaking, for calculating, that we use with the other students, is not going to work. And you can intensify it, you can do it more per day, you can do it more times per week, but those kids will not learn. If we look at fetal alcohol disorder spectrum, the kids that are actually diagnosed with fetal alcohol syndrome, fetal alcohol effects, if we look just at those kids, 1% are school-age population, pre-K through 12th grade, 1% are kids that are diagnosed on that spectrum. And if we look at that number of those kids, it's interesting. 50% of those kids will be in trouble with the law. 60% of those kids are going to have a disastrous educational experience. And I've worked with them, and I can tell you that they have terrible auditory memories. They cannot do the sounds of the letters for reading and writing. That you're going to have to go into that system and look at the neuroscience of the receptors and see how receptors actually work. And what that means is that instead of using sound-based programs that we have for literacy, we're going to have to use some of those visual motor ways to approach literacy. So a man by the name of Sodato, a neuroscientist, took a look at people who are blind, and he looked at what they do, what happens in the brain when they're doing Braille. And what he found was that the visual cortex lights up, which matches with what I have found working with these kids, and that is that I can use the motor, the shape of the movement of the hand, the shape of the movement of the mouth, the shape of the movement of being able to draw a picture or to sign. I can use the shapes, not the visual piece, the shapes, the movement, and basically create a literacy program for the kids so that they can learn. Their brains are different. They would have to approach them in a different way. Otherwise, we find that 82% of these kids at age 12 to 51 that have been diagnosed with fetal alcohol syndrome, and remember, not everybody gets diagnosed. There are a lot of people out there with fetal alcohol effects that have never been diagnosed. But 82% of those kids will not ever be able will not be independent. They will not be able to live on their own. And that's huge. 95% of them, 12 to 51, 95% of those individuals, that 1% of our population, will also have mental illness, okay? The one thing that I do want to say here about learning and about change is that the youngest kid I've ever worked with that has come to school drunk is 7 years old. Okay, so we're not talking 12-year-olds, 15-year-olds, 17-year-olds in terms of our programs here of intervention. And we knew that based upon his stories at school, based upon what he drew, that there was a lot of alcohol and parties and that type, late nights and you know, attendance problems, that type of thing. But the day that he came drunk, he was barely able to stand up. His parents dropped him off. And he also didn't have much oral language that day. And he had basically used an open container of beer to put on his cereal. So, you know, we thought, well, it's a one-time thing. But what we found, he was also obviously taken out of the home for a while and some other things went on. But what we found was they asked him to come back when he was 8.5 because they were having huge behavior problems with this kid. And he was different. He had mood problems, depression was setting in. There was all the slides that we saw about the brain, what happens, we were seeing with that 7-year-old. And at that point, he was just angry, and he was definitely what one of these folks has called in another speech a gulper. He had the genetic predisposition and the history. And if there was alcohol around, he'd go for it. And that was his whole day of what he wanted to do. Okay, so basically, we know we've got effects there. We know we have to change our programs because the brain is changed or has changed. All right, let's try another slide here. Okay, so educationally we need to recognize how alcohol and other drugs affect the brain and the development, how students learn. One more thing I want to say about the learning here is I'm not talking about learning styles. I'm not talking about preferences. I'm not talking about observing somebody and trying to figure out what style they use that they have been educated into. I'm talking about how those neuroreceptors, the way we see and hear, how they actually organize information from the outside into the central nervous system, how those become perceptual patterns. And we know those neuronal circuits that get larger and larger as you get up into the higher order of thinking for human beings. And what that means then is by the time I start using language, then I can start seeing myself and I can actually learn the concepts I need to learn to make the choices I need to make. So as we look at prevention and we look at intervention, we need to start looking at those learning systems and seeing how it is that we learn, and then apply it when we know that those learning systems have actually changed. And that basically means then that our intervention programs will be more pro-social instead of antisocial. Instead of telling students what they can't do, that we'll actually take the pictures, some of these wonderful images of the brain that the folks have brought tonight, take those pictures, draw our students into the cartoon of themselves, showing what they would do as they go through their day and what that would look like as they think about different ideas. We raise their cognition, their thinking, they're no longer into their own picture, other people are in their picture. And when we do that, then we can actually start making some changes in terms of the choices that they'll make. That's it.
All right, now there are ushers going up and down the aisle, so if you've got questions written down on your piece of paper, I see some folks holding those up, but let's pass those in. I'm going to start with just an opening question, if I can, to the full panel. It appears from the data that we saw tonight, and there may just be circumstantial from the slides that you chose, that we have a lot more data about alcohol use and abuse in adolescents than we do on some of these other drug systems. Is that true? And again, we'll need to sort of pass the mic along if you've got an answer to share.
I think you're on screen.
It is true. And it's because it's one of the most prevalent drugs that they use.
Clearly it's more prevalent. I sort of understood that. And yet I was thinking from the standpoint of the arsenal of tools that we'll need in the education community, it is the most prevalent, which means there's a certain sort of sarcasm on the part of the adolescent community about us trying to tackle that one. Some of these other drugs might be a little easier to get some traction, perhaps, because they're a little further on the fringe. I'm just wondering if there's any data that would suggest that possibility.
I'll answer again. The other, so alcohol is really interesting because from the way you framed your questions, children often have their parents drink, or even parents feel. The thing about alcohol is that it has the whole spectrum. Parents feel like it's safe enough that they can give it to their children with celebrations or religious events or even at a meal, all the way through the spectrum to it's flammable. You know, you wouldn't let your kid near 100% alcohol because one spark and it's, you know, on fire. So I think in the way it is so ubiquitous in our society at the moment, really, and the impact of public health make it have a higher precedence, if you will. I think the other drugs of abuse, as you say, there's going to be some tailoring. Clearly, methamphetamine is about the most toxic substance we know that people abuse. So it's frank brain damage. It's not subtle. It happens. There's a famous quote from marijuana, inside the office of the director in the 80s, saying, the only way you're going to get brain damage with marijuana is if a bale of it fell on your head. It's been really hard to unravel how it is that excessive marijuana actually alters behavior in a way that is debilitating to either a child or a person. So I think trying to unravel the different drugs of abuse rather than just clump them all together is the next, it really is the next frontier for us.
Just one comment about alcohol. When kids have experience with alcohol and then they're somehow denied that alcohol, they then go look for other things that produce the same kind of effect. And unfortunately, your garage is full of stuff like this. You've got toluene, you've got sterno, you've got all kinds of stuff there. And they start abusing those, they start sniffing glue, and basically, those are much more dangerous than drinking actually. Many of them are actually neurotoxic in their own right. But they're looking for that same sort of effect. So they look for these substitutes for alcohol, which leads to some very dangerous behavior. You sniff enough glue or enough carbon tetrachloride, I can guarantee you your brain has holes, so to speak. So,
I think coming back to base rates, though, again, we showed the slides. I don't know if any of us showed the slide I'm thinking of, but alcohol is the most commonly used substance in adolescence, followed by nicotine and marijuana. It's hard to disentangle the nicotine because those who use nicotine also use one of those substances most often. So I could have just as easily subbed out the slides that I showed you of alcohol on the brain for studies that I've been involved in looking at marijuana on the adolescent brain. There is pretty good evidence emerging that it does cause structural abnormalities, functional abnormalities in the brain, behavioral deficits, and so the reason we look at these two substances is because they are of greatest public health relevance. They are the ones that are the most used. And certainly, as has been mentioned, there are other substances that are far more toxic, but we've really focused our efforts on looking at those that are, well, most commonly used. Not altogether because it's just the most commonly used, but it's the easiest for us to study because of those rates. It's very hard to find a large population of people who use methamphetamine during adolescence. It's a hard group to study.
I'd like to make just one more comment on that, and that's the behavioral basis for addiction rather than just the pharmacological basis. So if a child or an animal or an organism finds whatever feeling that was, something it wants to repeat. So it could be Coca-Cola. I should have named a brand, but it could be a heavily sugar drink, right? The behavior that's built around that, it starts out, as one event and then a second event. And if there aren't competing or alternative reinforcers in the environment, then that behavior becomes more prominent, right? If there's nothing to compete with it. So if something happens, if a child takes a drug or shows up at school with a drug, and that state interferes, makes the reinforcing effects of being in this classroom negative. For example, it's not getting that positive reinforcement anymore. They're not doing well, learning, right? Or they're getting into particular social groups because they're perhaps banned from other social groups because of their behavior or just this like behavior. But there's a huge behavioral basis to addiction as well. And I'm not saying that these kids are addicted, but this is really in the very beginning of having this substance. The reason I got into this research is how can this substance come to control so much behavior? And those principles aren't related particularly to the pharmacology of the drug. Those principles cut across foods, social acceptance, just being able to get the car for the night, that kind of thing.
Kathy, it's interesting, you struck a chord with a number of our audience members here with the direction that you took that question at the end, with the sort of addictive behavior dimension of this, because clearly there's a neurobiological dimension, as most of you shared slides that pointed to that impact. But people are asking questions about a couple of other areas. I'm just going to hit them quick if we can. Sort of the innocuous one that we're all quite familiar with, caffeine. So what happens with early caffeine use? And how does that affect learning? And can they become addicted to caffeine? And is that problematic?
Well, certainly caffeine is going to heighten perception and sensation. and improve attention to a particular point. Almost all drugs have what we would call an inverted U, where there's going to be for whatever particular behavior or sensation or effect that you're looking for, it's going to increase to a point and then it's going to decrease. And so certainly with caffeine, if you have too much caffeine, you're not going to be able to pay attention because everything is catching your attention. But you might pay better attention than if you didn't have any caffeine and you're tired, for example, that kind of thing. But I also want to say that learning and learning behind reinforcement and finding a drug reinforcing is a neurobiological phenomenon. It's not that there is pharmacology in the brain going on and then some kind of learning around social acceptance. This is all neurobiological. It's all brain related.
Bonnie, you and Helen sort of handle the adolescent and education side of the house, so the other one, second only to caffeine, was video games. I know it's not the same category as alcohol and methamphetamine, but the look in their eyes sometimes makes you wonder, after they've been playing these for 4 or 5 hours straight. Is there something to be concerned there about the impact on the brain and brain development?
I get asked this question every time.
Well, we just wanted to round out that history, I guess.
So we don't know from a scientific standpoint yet what the impact of excessive video game playing does to the brain. I would say that it does lots of things. We know that the brain develops as a function of the experiences that we give it. So I'm sure that those kids that are rapidly playing video games develop very good fine motor skills and probably some good spatial searching skills and maybe even some good memory skills because those are all entailed. At the same time, you think about the rewarding effects of that game, and games vary quite a bit on this. What is it that makes it so fun? You know, what is it that keeps you coming back for more? That's driving the reward system in a way that is similar, not the same, to some drugs. I mean, it's changing the reward system in the brain. We don't know exactly how. We don't know the pharmacological mechanisms because you're not obviously, it would be endogenous. You're not putting something external in the body. It's changing endogenous reward chemicals in the brain, is my hunch. And that in turn is going to alter the course of development.
Yeah. But let's take a look, if we can, further out. Most of you were talking about sort of the slow recovery, if you will, and these were sort of measured in 100 days or a full year. How about multiple years? Is it possible to correct this early damage? And if so, to what degree do we get correction?
Broadly, I would say probably yes. I think one of the things that we don't know how to do well yet is at least from a pharmacological perspective or from even a learning and memory perspective, is to reverse those deficits that we see in the brain. If we could actually find some way to take that brain that looked cold blue, remember that one picture I showed? If we could somehow accelerate that process of the brain getting well again, presumably that would help. I also want to mention that the National Institute of Alcohol Abuse and Alcoholism now has a big center program which is focused on just that very question. It was just funded in December, is that right, Kathy?
The NAMBI?
Nadia.
Yeah.
Neurobiology of adolescent drinking into adulthood. I think I've got the neck. Anyway, this is actually centered at the University of North Carolina in Chapel Hill. And so they're just now beginning to do some animal studies along the lines that Kathy has talked about, where animals are passively administered alcohol during adolescent periods, and then they're studied as adults. They're beginning to collect cohorts of alcohol abusing 15 and 16-year-olds that they'll follow prospectively into adulthood, and those sorts of questions should be answered. We don't have as much data on that as you would like.
But there are some direct data on that out of Stanford University, where it's a lot like when we talk about multitasking. So we usually have a number of strategies to get from here to there. And if one route isn't available to us, we can go another. We can multitask. We can figure out ways how to do things. If what they found is in 20-year drinking alcoholics, even in abstinence, prolonged abstinence, they can do a task almost as well as the non-drinkers, but how much of their brain they have to use to get that task done precludes any other multitasking. So you can bring them in a laboratory and you can say, you know, what's your reaction time or something like that, and you can see it, but any complex task, this person then, that's the only thing they can attend to. And one of the really wonderful things of having the brains that we have with all this cortical material that we have to draw on is, and probably our evolutionary advantage, is that we multitask so well. So we can be shopping for dinner and thinking about tomorrow's agenda and on, and that's the part that really we don't think can recover.
Yeah, I just wanted to add something, that 33%, and by the way, that number of students, some people say as high as 45%, and I had a 5th grade teacher a couple years ago that looked at her students in a 5th grade class, and 9, 10, 11-year-olds, 90% of those students were not able to think beyond just themselves. And what's really interesting is if you're only thinking about yourself with your language, you can't really multitask. And that's something that we really need to understand, because that means that I'm in my own picture and I'm walking through that picture in a horizontal way, and I really don't see how other people affect me or how I affect them. So I can't think about other things that I would do. We also see in the language that they don't use the time for the future, which means that I can't prioritize and multitask into the future. So everything is from my present to my past, and there's some language studies about that type of thing. But basically, that multitasking function is one that comes with concrete to formal language. And if our kids don't have that, then they can't do it, and they're at risk to begin with for some of these behaviors. And then it's exacerbated, of course, as the brain is changed because of the use of the drugs, that type of thing.
What I find fascinating about that is that while there is not likely going to be a silver bullet here by any means, there's a lot of work to be done and some changing in programming. The fact that there could be long-term repair or, in a sense, diminishing of that damage impact, we still have the full impact on the learning side of the equation. So we might get the brain back biologically to a level that it can function, but we're way behind on the learning spectrum. And the possible silver bullet here, think of how valuable multitasking is to this younger generation. That might be a little wedge to explain to them just simply, you won't be able to do that very well if, in fact, that damage occurs early on. I don't know. I know how important it is to them to be able to do that.
Kids never think they're going to get old.
I know, they never think they're going to get old. Surprise, it really does happen. Panelists and Bob, thank you so much for joining us today for this program. What I want to do is just remind all of you that there's a reception following the presentation today where this discussion will continue. Dr. Thomas Green, Assistant Provost of the Community Graduate School, will be waiting down here to your far left. We'll escort you over to the terrace room where that reception will continue. Again, take a quick glance and grab some materials off the display tables out front. Thank you so much for joining us today. Educators, keep up the fight. Stay tuned. The University of Portland and OHSU, I suspect, will have additional programs of this kind to help you deal with challenges like this in your classroom environment.